Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show. Today bringing you a really fascinating guest involved in creating a better tomorrow for many people out there. Today we have the honor of being joined by what we call a biotech celebrity, that we have the Professor Dr. Thomas Rademacher joining us today, who is currently Chief Executive Officer and co-founder of Emergex, which is a really interesting company developing a novel nanoparticle-based vaccine technology to ultimately deliver synthetic viral fragments via microneedles and a really interesting skin adhesive patch, ultimately working on this principle of priming the immune system, immune T cells, and opening the door for something we discussed on past shows, that is sort of universal vaccines against some of these viruses that are extremely mutagenic, like flu and COVID. Professor Rademacher is a serial entrepreneur. He also serves as Emeritus Professor of Molecular Medicine, University College of London, widely considered one of the founders of this industry from the early 80s, having been involved in many of its disciplines, from recombinant proteins, monoclonal antibodies, the kind of spooky area, glycobiology, which we'll be getting into. And as an academic scientist, he has hundreds of publications, over 50 patents, 19 of which are in the area of nanomedicine. And he also, in addition to his expertise there, has been a pretty much vocal expert in the area of fetal-maternal medicine, having produced a couple dozen publications related to preeclampsia. And he was also co-founder of the field of glycobiology and the Glycobiology Institute in Oxford, and ultimately Oxford GlycoSciences, which was spun out of there back in 1998, which achieved a fascinating valuation of the London Stock Exchange. We're honored to have him with us today. A lot to get into. Professor Dr. Thomas Rademacher, thank you so much for taking time to come on the show.
Thank you very much for requesting to interview me, and your list of topics takes me back quite historically, I have to say. I sometimes I say to people, I was on some of the first papers that describe the word CD4 complement, and it kind of scares me that how far back some of these goes, but I've had the great privilege, I think, of being in some of the great academic institutions of the world through some of the most exciting things that one could imagine. I mean, during my years, going there, monoclonals were discovered, recombinant proteins. You're right back to the very first biotechs of Biogen, Genentech, and of course, Oxford GlycoSciences was the very first biotech out of Oxford University. And so one nowadays see these things as normal, and in those days, your academic colleagues, there was a real split between the people in the commercial world and in the academic world, and the crossing over, it was very, very difficult at that time. So anyway, I've had a really a varied experience, and I have to say, what I'm doing now, I look at as if I hadn't done all of the things that I had probably not done, I wouldn't be doing what I'm putting together now, because what I'm putting together now and we're doing, you couldn't conceive of in the 1970s and 80s. You just couldn't conceive that you could do some of the things that are done. And of course, we didn't have all the, I'm going to say it, that we didn't have all that modern instrumentation, and it wasn't, and you couldn't buy everything then from Sigma. You had to make it yourself.
Yep.
So, but anyway, it's been a very exciting time, and I think, yes, looking at the things I've been involved with, they did have a central a theme going through those.
Absolutely. I was intrigued, because obviously your name is synonymous with glycobiology, but
Yeah,
I took a quick peek
yeah.
back to 1979 and at your PhD, which is involved in how copper affects lymphocyte maturation. So it all starts off some of immunology, which we'll be getting to in a little bit. But take us back to the 70s, if you
Yeah,
would.
no. I have to say, which is very interesting on that, is as I keep people tail peeping. I said, "See, have you ever heard of the network theory of immunology by Niels Journey and people like that?" And they go, I said, I met these people. I said, I
Right.
met Auerbach. I met the first people. I met, Fritz Bach and Robert Good, or in fact, I worked with Fritz Bach and Robert Good when they did the mixed lymphocyte reaction with the first tissue typing for tumors. I knew, Howard Temin when he discovered reverse transcriptase. In other words, I think probably I've worked and talked to probably more of the Nobel laureates than my years. I have to say that I am. But overall, yeah, I was given I had a great opportunity to. I had a mixed in those days. You did when you did double degrees. I did a medical degree, and then I
Yep.
did a PhD. But that was actually in inorganic biochemistry at the Institute for Enzyme Research at the
Yep.
University of Wisconsin, and that was hardcore mathematics and spectroscopy. And then I had the real great. Well, actually, if you look at a lot of my papers, I was on all of the original papers on the origins of 2D NMR.
Yep.
Because when I went to Oxford, it was all being done there by Ian Campbell, and they had in the basement they had the first sort of real first Fourier transform NMR, and of course Oxford Instruments evolved, and that eventually became Siemens, and all the mRNA instruments and things like that. I remember we started out with 50 hertz machines, and by the time I left, it was 1,000 hertz or whatever it is. So, I mean, it was a great opportunity, to do that. And of course, even there, when you look at my doctoral thesis, it was on B cell. I always hated immunology my whole life, even though I've been involved with it. But what's interesting is it was on B cell
Yep.
lymphoblastogenesis,
Yep.
which B cells had only been discovered. And it's interesting because I once asked a couple of students, what does B cell stand for? And they says, oh, well, bone marrow. I says, no, it doesn't. It stands for bursa of Fabricius
Mhm.
from the chicken. Okay, so you had to go back. It was discovered in the bursa of the Fabricius in the chicken. That's where B cell comes from. So, I mean, it's scary to think back on some of these things, and you can trip some people up. But no, I mean, I think when I was only supposed to go to Oxford for 1 year, and we decided that in the great tradition that it would be good at that time. I'd finished my doctoral work and everything else, and had a job back in the U.S. And I decided that would be exciting to go off to Europe for a year. And so in those days, it was very, very difficult to get work permits and travel and things. It wasn't, fellowships to go abroad were impossible. And so I put in for a bunch of applications, and I remember I almost threw it away, but I got a letter one day from that I had won a North Atlantic Treaty Organization fellowship to go to Oxford. And I just almost literally threw it away because I didn't realize what it was. And I was very, very pleased because I discovered that year there was still, they still had them then, what are called NATO fellowships, and they were given to 50 people in the United States for all disciplines. In other words, you could be history, philosophy, archaeology, whatever it is. And I got this to to go to Oxford, supposed to be for 1 year, and I never left. We didn't return. So I'm an American, but an expat at that sense. So I spent most of my academic career at Oxford, and then I took the chair of molecular medicine at University College London. It was time to go to a bigger city, I guess you might say. So, but anyway, the opportunities, and through that, I met, some of really the legendary people at that time, because they all tended to come through those types of institutions, whatever it is. And I actually had a couple of Nobel laureates that, when they were on sabbatical at Oxford, shared my office. I won't give their names, but I thought that was, I used to get called up that he's going to be here for a while. What can you share your office? So anyway, I guess that was that. So glycobiology, I think, is very, very interesting. I'm glad you asked that question, because it was sort of I just got into Oxford, and R.R. Porter, along with Edelman, but R.R. Porter at Oxford just got the Nobel Prize for the discovery of the immunoglobulin molecule. Yep. And of course, we forget these things people didn't know about. Even in the 1970s, they didn't, what was an antibody molecule? What was its structure, or whatever it was? And so from that, of course, he got the Nobel Prize by saying it's made up of two pieces, or a couple of pieces, which is rather interesting. So the race was then on to do the X-ray of it, because nobody had ever done, nobody knew what it looked like. Right. And of course, the foundations of most of biology is based on what are called the immunoglobulin domain. And so he was upstairs and headed a department at that time. And so I sort of, my project there, was my first year there, was to sort out the sugars on the IgG molecule. Now, what do I mean by that? It was the crystal structure. They had done X-ray on it, but they couldn't really solve it because sugars are very floppy. In other words, so they didn't know. So it's sort of what unless they knew what the sequence of the sugars were, it was difficult to actually construct the final X-ray. Of course, nowadays, as you know, you can do this in a flash in a machine, but in those days, this was very difficult. So but at that time, nobody knew what an end-linked carbohydrate was. Nobody knew. There wasn't a field of glycobiology. Right. Nobody had a clue what was there. So I set about trying to, figure out what was on the immunoglobulin molecule, such that the X-ray people, that was Huber and Diessenhofer, they eventually got the Nobel Prize for that, could interpret what's called the FC fragment, an X-ray. So to do that, I had to learn how to do carbohydrate sequencing. And of course, there was really nobody in the world doing carbohydrate sequencing except a guy named Professor Akira Kabata in Kobe, Japan. And so I won't go through a long story, but I showed up one day at his lab in Japan, and it's sort of an infamous thing, like, why am I there? But anyway, I learned sort of at least the state of that art. And you can imagine in these days, you're trying to do alpha, beta, I mean, you couldn't, it was just, there wasn't a big NMR machine that could tell you what you had or anything like that. So I spent time training there, and came back, and we revamped a lot of the stuff there in Oxford. And based on that, we sequenced the first sugars. It was actually on rapid IgG of an antibody. And getting a little bit more confidence with that, I was actually handed the actual FC crystal that the X-ray was done on. We did a special order because it was all radioactive sequencing in those days. A special order to have super hot radioactivity because we're dealing just with just a crystal. And we succeeded in working out and sequencing the carbohydrates on the immunoglobulin molecule. And every picture in every textbook, if you look at it now, when they got a picture of the IgG molecule in it and they got the sugars in it, those sugars were my sugars. Okay. So the importance of that study was the following, is that we discovered that the sugars are these carbohydrates, which now we're starting to be sequenced by other groups in the world. We're on the inside. And the IgG molecule remains today the only protein that has the sugars in the middle.
Mm.
So you have to sort of think about that. You got to see all these pictures of spike and all of these things and everything else. The sugars are flopping around on the outside. But
Right.
the immunoglobulin sugars were on the inside, which was why they had trouble solving the X-ray, in other words,
Mm-hmm.
because it was on the
Mm-hmm.
outside, they just flop around, you wouldn't see them, but they were on the inside. And there was two of them that were paired together. So we then, from that, working now being friends with the Akira Kabata in Japan, we set out, we started looking at just immunoglobulins from individual people.
Yep.
And there was this theory early on that there was something funny about the immunoglobulins in people with rheumatoid arthritis.
Mm-hmm.
They used to say, these people form immune complexes, so they do something, but nobody had ever been able to figure it out. So we set out with Akira Kabata to do a sequence, which was a huge amount of work today. I can't remember, 50 or 100 people or whatever it is. We had to isolate their IgGs and sequence the immunoglobulins from people with rheumatoid arthritis, juvenile arthritis, and we did a whole bunch of things. And from
Yep.
that, we discovered, of course, that the cause of the autoimmunity in rheumatoid arthritis was because the glycosylation of the
Mm-hmm.
antibody molecule in that disease differed. And so that was the first glycosylation disease ever discovered. It stands still today. And the idea, what we discovered was that these sugars, which were on the inside, normally, these end-linked sugars and in the world of glycobiology, they get what are called end-capped. That is, they get something called sialic acid or
Yep.
galactose on it. Okay. These things are recognized as self in the human being. So that just says, forget it, ignore it. The next sugar down, so you got three sugars on the end, sialic acid, galactose, and the next sugar down is N-acetylglucosamine. Well, N-acetylglucosamine is on the surface of bacteria.
Mm-hmm. Mm-hmm.
So if you have a sugar that ends up ending with N-acetylglucosamine, the body thinks it's a bacteria product.
Sure.
So that's why these sugars on the immunoglobulin molecule were on the inside, because a lot of them ended up with these N-acetylglucosamines. They had to be hidden from the bloodstream. They had to be hidden. But in rheumatoid arthritis, they had an increased number of them. And when these things bound into the joint, we found, into the collagen of the joint, they flipped out. And that activated complement and caused the inflammation in the joint. So this then became what was called pathogenic immunoglobulins.
Mm-hmm.
So
Mm-hmm.
immunoglobulins that contain these IgG bacterial-like sugars. So why was that important? Well, monoclonal antibodies were just about being discovered then.
Right.
Now, if you made a monoclonal antibody with the wrong sugar on it, you had real trouble. You could see, because it was pathogenic. The body recognized it as being a piece of bacteria. So in the early days with the monoclonals and everything else, there was a rush on then to engineer the early monoclonals with the correct carbohydrates on. So this was, I like, is my claim to fame. So anyway, so how do you do that? Well, in those days, we didn't have all these fancy things. And what we discovered was that if you made the immunoglobulins, in those days, they were making them in cell lines, of course. So, and they were making them in, they don't make them like they make nowadays, but they try, if you make them in yeast, it's even worse, because then they get yeast sugars on it. So what happens is these poor antibodies or monoclonals always carry the characteristics of the species they've been made in. And so what we did is we, by chance, discovered that Chinese hamster ovary cells, CHO cells,
Show. Yep.
put on human glycosylation. So all of the first monoclonal antibodies, you will say, actually were made in CHO cells so that they had the correct glycosylation. They were silated in galactose, whatever it is, and they were not pathogenic. And that allowed the early monoclonals to be used in the early therapeutic days without causing massive reactions. And eventually, of course, so all monoclonals to this date are always engineered now not to have these type of sugars on. But without that understanding of this area, monoclonals would not have been a therapeutic agent, because we want them to go in and bind, neutralize tumor necrosis factor or a cytokine, but we don't want them to then set off the complement system in the body and have a thing. So that whole field evolved from this this setting. So it's interesting how the glycobiology became a really very, very important, because the same thing then occurred in recombinant proteins. In other words, oh my goodness, if I make a recombinant protein in a mouse, it looks like a mouse.
Right.
And as you know, even now today, that's the problem with giving the pig heart to a person, whatever it is. It's the sugars on there that you recognize. So they have to engineer these things off. So I think we were probably, I think we've probably sequenced more carbohydrates when I was at Oxford than any other group in the world. We did all the interleukins. We did, there was things that have never been before. We did plants. We did dictyostelium. We did all of the leishmania. So we really classified all of the whole field of biology, if you want to look at it that way, during about a 10 or 15 year period. And that, it all stands today. But of course, now today, when somebody wants to, I think there's probably maybe one or two people in the world, if there still is, that could actually know how to sequence a end-link carbohydrate or complex carbohydrate. Because now what you do is you buy the standards and you run them on an HPLC machine and you match up the standards, okay?
Right.
You don't go in there and say, is this an alpha bond, or is this alpha galactose, or is this beta galactose? And how is that linked to, I mean, these were very, very complicated structures. And of course, we then worked up the biosynthetic pathways, the high mannose pathways, all of that, and then how they were made and in the post-translational thing. So yes, and then we finally wrote it all up and we called it glycobiology. I think that was in 1980 something another, in Annual Reviews of Biochemistry. And that was the first time the word glycobiology had been used. Before, nobody had put it together. And at that time, we defined the field, and we defined all of the components of the field: glycoforms, glycotypes, in other words, all of the terms that are used today came from that. Here is the description of what glycobiology is. So that was a very important, and you can see therefore the Oxford GlycoSciences is, of course, was therefore a logical spin out at that time, as you can imagine there, because then if you looked at it, well, once you understood the glycosylation, obviously there's going to be compounds and drugs that inhibit glycosylation. So that was then started on the fact that there must be, and we knew a lot of antibiotics were doing this, and we then discovered the nitromycins, which are sugars with a nitrogen in the ring, and to this day they are still products developed by that company are still used today for a variety of Gaucher's disease, a number of diseases. They're still given today that came out of that. The first HIV drug, which was called Oxades, whatever it is, for whatever reason, which we developed, was actually a very, very good drug to upset the glycosylation of the HIV virus. And that was taken into clinical trials, but then didn't progress because then they discovered all of the new, the protease inhibitors, whatever it is. But I'm pretty sure that was the very first prototype drug, and that was taken up at that time by the Monsanto Cereal Corporation, and it made kilograms of the stuff. But as I said, it was just taken over by other drugs, whatever it is. But it stands to this day as a very good HIV drug if one wanted to have that. So the next thing that came out of it, working our way forward here, of course, is that we had there was unbelievably in the 1980s, everybody thought that all proteins in a human cell had what is it, membrane sequences that went through. All proteins on the cell surface had a sequence that went through the membrane, and that is how all proteins were attached on a human cell. That was in every textbook, everywhere in the entire world. And working with a guy named Alan Williams, who discovered the first, it was called Thi-1, which was the first of the I would call it CD. Now you have CD28, CD4, CD8, whatever it is.
Sure.
So the very first, I guess, surface antigen ever discovered was called Thi-1, obviously for T cell, and it was the first marker for a T cell called Thi-1. And we got involved with that because we were going to do the sequencing of Thi-1. We wanted to look at the carbohydrates, and it was a single immunoglobulin domain. And so we did that, and then something was funny because it didn't have a hydrophobic membrane tail, but yet it was a membrane-bound protein. And this was one of the first membrane-bound proteins ever physically isolated. And we went about to figure out what it was, and we discovered that it didn't have a hydrophobic tail. It had a big long name called a GPI anchor, a glycosylphosphatidylinositol anchor. And all of a sudden, we discovered that most of the proteins on the bloody cell were not anchored by amino acids. They had these GPI anchors. So we went on and discovered the GPI anchors. And of course, they're very clever because then you have a cell surface protein that can get clipped and it can be released as a soluble form, or it can stay on the surface. But what's fascinating is we then discovered the entire parasitic kingdom only uses this. So if you go into Leishmania or trypanosomes, or we did all of these things, so we did all of the parasites, malaria parasites, they only use these GPI anchors to attach their protein to the cell surface. So we wrote a wonderful article in, I think it was 1984, that in 1984, you can still discover that everything in the textbooks prior to that is actually wrong. So and then that exploded, and that was a huge field.
Yeah.
But moving on then to how we're getting there. So I got very involved with maternal fetal medicine, and preeclampsia. In other words, in the West or the North-South divide, it has an incidence maybe 3 to 5%. Now, I think that's a lot. In other words, 5% of all pregnant women can come down with preeclampsia. Now, in modern Oxford, here where I live, of course, if you have any signs of preeclampsia, early proteinuria, or hypertension, whatever it is, you get thrown into the John Radcliffe Hospital and you get $1 million treatment, and they'll deliver you at 27 weeks or whatever it is. But on the other half of the world, it's the leading cause of morbidity mortality. In other words, it is a massive, massive problem. I was really very, very fascinated with preeclampsia because when you think about it, in the old days, if you caught preeclampsia before modern therapy, it's not just the baby that died, it was the mother and the baby, which was rather interesting. You couldn't, in other words, it even, so it really is, it was called toxemia of pregnancy. It's a wonderful way of looking at it. So I got involved with it because I was an immunologist, I suppose, in the hardcore in the end. And we did an awful lot of work looking for the toxin. because everybody knew that as soon as you delivered, or the woman delivered, she was okay. So the woman would be very, very sick, and then she would deliver, and within days, she'd be, she'd much better. And sometimes, they could progress. These women could progress to eclampsia, which epileptic fits.
Mhm, mhm.
So we knew there was something in her blood that was there, and everybody had searched for the toxin, or that was called the toxemia of pregnancy, or preeclampsia. So when we started looking at that and finding that, we found a substance in the blood that, to our amazement, had structural similarities with the GPI anchors, which we had discovered, which attached proteins to the thing. And in fact, they were the structures. And we called them the inositol phosphoglycans. So again, carbohydrates, but they were sort of like the middle segment of a GPI anchor. And we then discovered that these were rather interesting second messengers. As you know,
Right.
there's a lot of second messengers around, and that the fetus utilizes these inositol phosphoglycans instead of insulin. Now that nobody knew that. In other words, a fetus lives by what's called histiotropic metabolism. That is, it doesn't, it lives, it doesn't, it's totally different. It eats, its food source is sorbitol and things like this.
Mhm.
So these molecules we discovered that were probably poisoning the woman were present in huge quantities in the amniotic fluid. And it took only, a rational thing to say, well, some of them might be leaking out,
Right.
and if they're going to leak out, you're in trouble.
Yeah.
So it turns out that they also then have structural similarities with endotoxin.
Ugh. All right.
So
Here we go.
basically, the woman was being poisoned with a natural form of endotoxin, if you want to look at it from that way. In fact, they cross-reacted an endotoxin, a bit. So this was a very, very important aspect of that. And of course, I got very interested in the whole epidemiology of preeclampsia, because originally preeclampsia was said that it occurs during the first pregnancy, and it doesn't occur in the second and third and fourth. It was always called prima partum, the first pregnancy.
Mhm.
Then some people we were working with came up, and they had all of these cases. Women got preeclampsia on their second, third, fourth, you know. And when we looked at it, they all came from matriarchal societies. Now, what I mean by matriarchal societies, I mean societies where women tend to have change partners all the
Mm.
time. And of course, what then evolved was, it was on change of, that is, if you stayed with the same partner, it was okay. But if you changed partners, you could come down with preeclampsia. So you started all over again. So it became the peri-paternity. In other words, first time. In other words, if you change partners, you're back.
Yeah.
So we put this all out, and of course, as you can imagine, this caused absolute chaos.
Sure.
So you imagine now you're at a dinner party, and somebody across the dinner party, this lady is sitting there, and she says, "Yeah, yeah, yeah, I had preeclampsia after my third child."
Mhm.
Okay, now you go, okay, all right. Really? After your third child, So anyway, this got to be very, very, as you can imagine, very, very controversial. And, there was this joke that nobody knows who in Scotland anyway, but I mean, it was, so anyway, but to this day, it stands that, yeah, I
Right.
mean, I'm sure there are cases where the women have stayed with the same partner and had preeclampsia later on, but it's a rare event.
Sure.
Let's put it that way.
Yeah.
So
Understand.
I thought that was I thought that was a very interesting one. So you can try that out at a
Yeah.
dinner party when somebody said, "Oh, I had preeclampsia," whatever it is, and yeah, and who's the father? So
Right.
any,
Right. Right. Right.
uh,
No, I
so
don't.
anyway, the long and the short was, therefore, uh, we really wanted to do something about this, and the we looked at this, and yeah, there probably was in preeclampsia a leak. and this stuff was coming out into the blood. We could measure it in the urine of the women. It went through the blood, whatever it is. And from that, we developed a diagnostic that would, once the woman started to leak, we could measure it. And we knew if she had, and we could measure it in urine, so it was very easy. And so every couple of, if you measure it 2 weeks, couple of weeks, and she's still positive, she was going to get preeclampsia. So
Mhm.
that became very important because you have to think about a therapeutic for preeclampsia. You can't go out and treat all pregnant women with something because 3 or 5% of them might come down with preeclampsia. In other words, you just can't do that. And so you had to have an indication of not only the at risk, but really, if you're going to do a clinical trial, you really needed to know they were going to come down with preeclampsia before you put in an intervention therapy. Because you
Mhm.
have to think that this is benefit and risk. It's a pregnant woman. We can't tell if they're going to get preeclampsia until about 18 weeks. That's when the placenta will start to leak. So it became, unfortunately, there was a drug that we thought would be very, very useful. We convinced the pharmaceutical company to run clinical trials on it using this, and COVID hit.
Yeah,
And a year into it, they gave up because they couldn't recruit. You couldn't recruit people
sure.
or anything in clinical trials. So it never made it never made the clinical trial bit. And that's one of my great regrets because it is a very, very, very serious problem in the Southern Hemisphere. And without, emergency treatment, the mother and the baby, will die. And then you can get into the whole issue of why we have what is preeclampsia, and that gets even more interesting. Is the There was a study done that it all has to do with degree of cohabitation.
Mm.
And in other words, such that the chance, if you and a partner have sex for the very first time and she gets pregnant on the very first time, the incidences of preeclampsia are very, very high. And on the other hand, if you cohabitate, by the time you've been sleeping with your partner for a good old 7 or 8 months, the chances of getting preeclampsia go down to what's called a compression rate of around 3%,
Hmm.
whatever it is. And so people have thought a lot about why the biology of this is. In other words, is this the human nature telling us we really need to be with someone for a period of time to know we really want to have a child with them? In other words, of looking like that. Or is it somebody else once prescribed? Is it a natural anti-rape response? Because, in other words, if a woman tends to be raped and get pregnant on that rape. In the old days, she would probably have had, there was a high probability of preeclampsia. I mean, we will never know, but the incidence of preeclampsia has not changed in the centuries. It stays there. So it's not like something that it can be it's not like we get rid of the people who get preeclampsia. It
Right.
seems to be a natural interaction between the father and the mother. There's some combinations that just don't work, and the body knows they don't work, either by cohabitation or whatever it was. And of course, then there was the famous Dutch study by my best friend Gus Decker, who couldn't had to had you couldn't resist is could you get protection using oral sex? I mean, that's the thing you wanted to know, I think, and whatever it is. So when he published this paper getting a hold of that, doing only in Holland could you do the study, of course, and of course, for many times it was one of the most highly cited papers in all of immunology. And yes, oral sex does work, by the way, and gives you the same protection, by the way, in case you wanted to know
Hey, now we
that
know.
fact. But it was it's one of the landmark papers in immunology.
Really, no, no, really, really
Uh,
interesting.
so,
No, it's yeah, you've been you really been on the forefront of a lot of stuff,
uh,
Tom. I appreciate that that extensive background.
yeah,
And now so we talked immunology, we've
yeah,
talked glycobiology,
yeah,
we've talked foreign,
Yeah.
microbes and
Yeah.
how they present. Let's bring it all together
Sure.
in what you're doing now, because, and
Okay.
again, there's some areas here that will need a little hand holding on, but Emergex is a fascinating story in its
It's
own right. Um, let's start off first with, what you introduced when we go to the, and we'll put the link to the website in
yeah.
the bio of the show, this principle of CD8 positive T cell priming.
Right.
And again, I personally, I think the immune system is more complicated. I understand the central nervous system better. So a little hand holding there, but we have this pool of these
Sure.
CD8 T lymphocytes. They're an important part of the adaptive immune system. A lot of the times they're resting and hanging out, not doing things until they're presented with antigens from bone marrow. Um, Introduce the
Yeah.
principle first of T cell
Yeah.
priming.
Okay. So the we very clear here. So the body evolved probably evolved the CD8 T cells to deal with viral infections. That's what they're there for. The CD4 we talk about all the time is great for antibodies. It does helper and things like this. Just put that
Hmm.
aside.
Hmm.
So and the other way of looking at it is antibodies go after free virions, ones that are floating around. So they got
Hmm.
to
Hmm.
be floating in your bloodstream. They got to be free. They got to be somewhere around. And most viruses are quite smart. They don't like to float around. They don't have what's called a lymphohematogenous dissemination spread. A lot of them simply spend their entire life inside of cells. They go from one cell, A cell to A B to cell C. So they live an entire intracellular life.
Yep.
So what happens is let's just take an example here. Can we get to T cell priming? The most of the world's nasties are RNA viruses. Okay, this is HIV, flu, polio, yellow fever, all the things that we think, COVID, all the things that monkeypox, all not monkeypox, that's a DNA, but most of the things we think that are going to get us in the
Yep.
end are RNA viruses. Now, RNA viruses are very small. They're little tiny things. The dengue virus contains just 6 proteins. Yellow fever, I think, is 6, whatever it is. They're very, very, very small.
Hmm.
And because they're RNA, they have no correction mechanisms. So DNA, we correct. RNA, we deliberately do not correct. So if an RNA virus goes into you, as soon as that RNA gets replicated, there's a probability at every single base of having a mutation. It cannot reproduce itself the same. It's actually impossible. It turns out it's the complete opposite of DNA.
Right.
So what it does is it makes a cloud of viruses, and they're called quasi-species. So I used to say to people, there is no such thing as the Wuhan strain. The Wuhan strain was a mathematical construct. What they did is they do sequencing. And if you can imagine, I'm going to sequence a cloud of viruses.
Hmm.
I have to add up the most likely position at each point, whatever it is. And so the consensus sequence was simply the most likely amino acid that occurred the most times at any given point. So it was the sum of all the quasi-species. So when they made the spike protein against the consensus sequence of Wuhan, that spike protein probably never existed. It never did exist because it was a mathematical sum of all of the quasi-species of the clouds. So why do we do this? Why is this done? Because there's not enough information in an RNA virus to make it pathogenic. In other words, it's what it does, it makes friends. Okay. And the best example of this is polio. In other words, they've done some wonderful studies on polio. Polio is an RNA virus. And then you say, well, in other words, it makes friends. Now, unfortunately, sometimes those friends can cause a problem. So, as you know, with polio, something like only 0.1% or something of the kids that caught polio actually had neurologic complications. That's because one of those friends in there figured out how to get across the blood-brain barrier or how to get into a nerve. But 99.9% of the time, that cloud didn't contain that particular subspecies. So they work together cooperatively. Okay, it's like it's like the herd of, it's like these you always see these killer whales or whatever is working together or whatever it is, and very people understand that there it's like these things are all quasi-species clouds, and that's what they do. So if you then think about an antibody's task of trying to kill off a cloud, well, you're swatting one, and then you got another one that's
Right.
different than another one that's different.
Sure.
So the body had to come up with a clever way of doing it. And of course, it decided, well, I got to kill the cell that's infected without looking at the virus at all. In other words, because if it looks at the virus, it's stuffed. It's had it. Okay,
Yeah.
because there is no such thing as Wuhan or Omicron. There are, in other words, when Wuhan became Omicron, but remember, it's just the difference. It was just different numbers of these. The frequency of these things just changed. It was
Yep.
all so. It's just the frequency change. That's all it
Okay.
did. That's why it could morph so quickly. So the Achilles heel, therefore, is that the RNA virus has to go in and make lots of friends. So it's got to make a cloud, and then that virus, that cell will explode with all of those friends. In other words, a million variants, whatever it is, then get spread out into you. It's got the Achilles heel, therefore, is it takes time to make all of those variants. In other words, it takes about 18 to 20 hours for the first virions, intact virions, to come out of an infected cell. And that's what's called the eclipse phase. So the eclipse phase is the time from infection to the time you produce virions, which can go on and cause transmission. And in this case, we're talking transmission within the body after you've been infected. Now, in general, people don't, they sometimes forget, you ask, how many viruses does it take to infect somebody? It's got a weird name, the bottleneck. And it's the complete opposite of what you would think. So the bottleneck is how many viruses I have. If I have flu, how many viruses do I have to spit out, or aerosol, to pass from me to a recipient, from donor to recipient?
Right.
How many? That's how many do it. But you have to understand what's happening is if only one of those goes, you're only getting one of the cloud. Okay, so
Right.
I may have a cloud in me because I've got flu, but if I put only one across, then I'm only sending one member of that, and it's got to recreate the cloud. And in general, the flu, COVID, eventually, these viruses eventually go to a bottleneck of one, which really means you just have to have one virion to infect you, and it can grow up from that. Okay, just one needs to make it. That's all that has to happen. And in general, interesting, viral infected cells are sort of like the egg and the sperm. When sperm goes in, it doesn't allow other sperm to go in. Same thing kind of happens with a viral infection. Once goes in, it doesn't want anybody else to come in, and it tends to take over, which is rather interesting. So now the thing is, as soon as that mRNA, that RNA virus goes in, the first thing it does, it has to make some things to make the building blocks. So it isn't necessary. It doesn't replicate to begin with. It's got to make some proteins and things because it's an RNA virus. So obviously it can make proteins directly. It doesn't need to go from DNA to RNA. It can go it's straight to RNA. And as soon, within seconds of infecting the cell, it starts to make what's called the pioneer phase. It starts to make peptides. Well, in that sense, what it's called is starts to make viral protein. And that viral protein gets chopped up into peptides and gets put into the class 1 HLA molecules of the infected cell. Now, the class 1 HLA molecules are the things that tell you are you. So they contain all your self peptides. In other words, so the body knows all of those peptides on there. It's sampling everything in your human cell that is you. That I don't attack. But as soon as a viral peptide appears there, which can happen within 30 seconds or a minute of infection, that cell is targeted for death because it's foreign. As far as the body is concerned, it's now foreign. So now there's a race. Now there's a race because this viral infected cell has been identified or is going to be identified as foreign, but it's got to make a bunch of its friends before it gets annihilated. Okay, now you see what I mean?
Right.
So in a natural infection, so what happens is that information gets passed on to the immune system. And I think I won't go into it. It's very clever how that's done, because if you get a dengue, you have to ask, your lymphocyte, your lymph nodes are not normally infected. You get infected in your nose or your skin from dengue. There's information transfer from of this infected cell. Actually, the class 1 peptides move to the immune system. And the other fascinating bit is that you and I and everybody have got to throw the dice on T cells, because what we want to do now is generate a T cell army, and they are the type CD8. Let's just call it that. And 8 means nothing other than it was the 8th CD that was done. There was a CD, 7 and 4. But CD8s are what are called killer T cells. And they are things that if you have around, will annihilate this infected cell because it thinks it's foreign. In fact, what it does, it thinks it's a transplant. It's the same mechanism. If I tried to put some of my liver into you, my immune system would say, wait a minute, that's foreign. That's not me.
Yep.
And it would go after that and it reject it. So the attack is no different. So the viral attack of the CD8 T cells is essentially no different than what's called the allogenic response, which you have tumor rejection. So now we get to the interesting point. Now, you have a thymus, and T cell, in this case, stands for thymus. Nothing fancy like the bursa fabricius in a chicken, but it stands for thymus. And up to about 7 or 8 years old, your thymus is called throwing the dice. It's making T cells by random chance of different specificities. Okay, something like 10 to the 11th of them, it makes these things, and then it stops making them. Now, this is very conceptually different from an antibody response. An antibody response can adaptively make something at any time. It's presented with benzene, and it'll make an antibody against the benzene. But the T cell system, you're fixed. You got to throw the dice. So you've got these, and within that pool, you better hope that you've got some that might react with dengue, or some of them that might react with flu, because if you don't, you have a hole in your immune system. So they're pre-existing. So that's important, because if they're pre-existing, you don't need to generate them. So as soon as they think there is a danger out there, as soon as they think, so I've got now ones against dengue, one of these T cells against dengue, and I get bit by a mosquito, that information gets to these guys. They immediately start to explode and expand. 10,000-fold in a couple of days. And they reach their peak about day 8. And that's when you get the runny nose. Because about day 8, your T cell, that's when you feel like crap, because your T cell system is now moved in and is killing that. Up to that time, you're pretty okay. In other words, and so the race here is between the virus generating all its friends in the first 6 or 8 days before your T cell army comes in and annihilates it. So that's the race. We normally win the race. We normally win that race because we have other mechanisms to slow the virus down. This is innate and gamma, things like that.
Yep.
So therefore, T cell priming is fundamental, because you can then say, well, I'm taking a chance with the race. So why not have the T cells there before the infection? Now, this one guy comes in. This one guy comes in my nose. This one COVID-19 comes into my goblet cell in my nose. He's got a problem because, I have now got 0.1% of all the T cells in my body, sitting there waiting to kill this guy because I've already turned him on. So what we do is nothing other than create the T cell army from your own army that you have. You can't generate something new, whatever it is, but you can turn all those guys on, and they're all now primed. They're all what are called, and I'll get into what we mean by that, but they're all ready to go. And so now, as Fauci used to say, I want a COVID vaccine that gives me a runny nose. Because what he's really saying is that's what you want. You want cellular immunity. So, I get that infection in my nose, and I want the T cells to come in, and of course, they'll create, snot and things like that. So what we do is effectively, therefore, activate your T cell army, the one that you have naturally, and what the one you would use if you came down with dengue or COVID, whatever it is. You can't, you got your throw of the dice, but these guys at least are there on day zero. In other words, to do that. Now, if they kill the infected cell before it makes any friends, it's called an abortive infection.
Okay.
Perfect. You got infected,
I like
you
that.
killed it, it didn't produce any friends, you're fine. That's how the live attenuated vaccines work. That's how polio vaccine works, yellow fever, measles, mumps, whatever. All we're doing is a version of a live attenuated vaccine without using live virus.
Mm-hmm.
So if you want to encapsulate what we do, is the trick here is to make the body think that it's had a dengue infection or it's had that with something that isn't living. Okay, so with yellow fever vaccine, I got to give you actually get a very low grade yellow fever. That's what you do. It's live attenuated, but we'll go into what that means. Okay,
Right.
so what's interesting is nobody been able to achieve that. So because, let me give you the example. If I take live yellow fever and I vaccinate myself, my body will respond in a particular way. It'll make a CD8 T cell response. It'll make this army. It'll do all the kinds of things. If I so much as sniff on this yellow fever vaccine, if I sort of heat it up a little, if I put a little alcohol in it, whatever it is, it may look, I mean, I can do whatever I want such that it doesn't replicate. And I vaccinate myself exactly the same way, it doesn't work.
Mm-hmm.
So the body knows what's alive. And the question, therefore, is what is the body recognizing as live or not alive? Because it has to recognize that before a lot of things happen, if you see what I mean.
Yep.
So it was generally thought that you could not reproduce the live attenuated vaccines with something that's dead. Okay. So if you want to say something weird, we're saying we know how to make the body, we're tricking the body into thinking what we give is alive.
Mm-hmm.
Now, and the trick, it turns out to be all in the way these vaccines, if we want to do it, they're really not vaccines. If you can imagine what I've just told you, they're programming. Okay, so basically, we're going to tell the body to turn on T cell A29999. Body has no choice. It's just going to do it. It doesn't process anything. It doesn't do anything. We're basically software engineering the cell. So there was two aspects then. You needed to know what to engineer, what the software, what is, and it turned out we then discovered that each virus has got a unique signature. Dengue is dengue. The body knows what dengue is, interestingly enough. If you catch flu, the body knows what flu is. This is all way back probably in our genes somewhere. So it knows actually how to deal with these things. So what we do, therefore, is in order to recreate this live attenuated vaccine, we have to know all the signals a live attenuated vaccine would give. And therefore, what we have to do is we have to infect human cells in a laboratory, whatever it is, infect them, and then we physically have to extract and sequence all of the signature. So we get what's called the dengue signature, which is maybe 60 peptides. There's the flu signature, and then there's the yellow fever signature. Each signature is unique to that virus, interestingly enough. So now we know what we want to do. We want to tell the T cells against these things, turn on. We want to turn those on.
And this signature is what you refer to as sort of this ligandome.
That's right. It's
called the
ligandome.
Of okay.
That's right.
All right.
That's right.
It's a library. Bring that in now, and because there's a second part of this which comes back to glycobiology and these carbohydrate nanoclusters. Yes. So walk us through this now, because this is
okay.
So elegant.
There was two. Okay. So now you have to ask. Well, now I got to make the body think it's alive. Okay. Now you see where we're getting at, and this actually gets back to what the famous word. I know it got MIT. I think I can't remember Harvard and somebody else in trouble, Pennsylvania, over the word contextual. Okay, I think they got in real trouble over the word contextual, and a couple of presidents of universities lost their job over the. I think on that. But anyway, the uh, the the body sees this ligandome, and it has to see it in what's called a contextual nature. It has to see it in a manner, somehow or another, in which it deems this stuff it's alive, that it's come from a live virus, put it that way. So this software, or these, excuse me, I don't know, it's hardware or software, this ligandome, the things that we want to deliver, they physically have to be delivered, are put on a quantum crystal. Actually, it's not actually a nanoparticle. They're put on a quantum crystal. And it's a 104 atom crystal that we discovered. We also discovered that, I have to say, came up. We also did the first nanomedicine company, which we haven't talked about today. But we discovered these molecules, and they're a very unusual form of gold. They're an artificial atom, in effect. And they're purple, so they're not gold. But they're extremely minute. Okay, we're talking, smaller than, they're about the size of 10 sugar atoms, whatever it is. So it's a crystal, and it's called a quantum crystal because they have very unusual properties to that. But you can do chemistry on them. Okay, and the chemistry that you have to do on them is all in the trick. That's what the vaccine is. So if you can imagine, they're not actually a ball. They sort of look like a drum, but the easiest way to describe them is they're a very small ball of gold. And we have to coat them in a way that the body thinks they're coming from something alive. Okay. Now, the trick is, if you can imagine a ball, and you can imagine the rings of Saturn. Okay, easiest way to look at it. Can you imagine? So what we have is two things that we coat these particles with, and they're put on as rings. So there's a ring of A, then a ring of B, then a ring of A, and a ring of B. So they're not a mosaic. Imagine them is they're stripy. They're called stripy. So it's a series of rings. So there's, in other words, so imagine a ball in which you've got a series of rings. Now that ball, we could have put these two things on as a mosaic. In other words, if you imagine that. In other words, they're just sort of spotted around on the surface, right? Or we could have put all these things on one top of the ball and all things on the other side of the ball. Okay, but we didn't. We had to work out how to put them on in rings in a synthesis. Now we're talking about, really, when we discovered to do this, this was beyond, this is before people could do individual syntheses and things like that. So why do we want to do that? Because there's a very unusual property of putting things on in rings, is they have, it creates a pattern. Okay, so what we actually do is create a pattern. And in the human body, there are what are called pattern recognition receptors. So they don't recognize chemical compounds per se, they recognize the patterns of which the compounds are arranged. And it turns out that bacteria and a lot of things in the nasties world kingdom have got the cell surface is put on as like ribbon-like structures. So they have a pattern. So when the body recognizes bacteria, it does it through what are called pattern recognition receptors. So it doesn't recognize it because it's N-acetylglucosamine or this, but it recognizes this pattern of molecules, not the actual residues themselves. So what's on the surface of this molecule is basically a pattern that appears on the surface of most bacteria. And the body then goes, oh shit, that's not good. So the body never sees our crystalline gold. It sees this. And going back to carbohydrates, pattern recognition molecules are carbohydrates.
Yep.
So the surface of this is there's a ring of N-acetylglucosamine, then there's a ring of galactose, then there's a ring of N-acetylglucosamine, then there's a ring of galactose. And that exactly mimics the peptidoglycan surface of a bacteria. Now, why is that really important is because if I take a gold nanoparticle that people talk about and I throw it into serum or plasma, everything's going to bind to it. You form it's a colloid, so things stick to it. Okay, in other words, albumin will stick to it and everything else like that. But we're trying to deliver something on the surface of this particle. And believe it or not, when you create this pattern, nothing sticks to it. It's a Teflon surface.
Hmm.
Absolutely. You can throw it into blood, serum, whatever it is. Nothing binds to it. Albumin, cytokines, nothing in blood binds to it. But my God, does it glue itself to your old dendritic cells and the antigen presenting cells.
Nice.
So creating the pattern recognition targets this thing, believe it or not, to the cells that are designed to go out there and look for all the baddies in the world. So onto that little tiny particle then is placed the peptides from the ligandome.
Mm-hmm.
So what happens is we inject them in, nothing else binds to it, and they get taken up absolutely specifically by the dendritic cells in your lymph nodes or in your skin or whatever it is. They unload these this ligandome thing. It goes into the dendritic cells and it turns the T cells on. So a lot of physics involved in that.
Absolutely.
So and then there's two other last pieces that I think are important.
Sure.
The peptides themselves are going to be chewed up and they're going to be food. So you're now trying to deliver a code that's going to be degraded within 10 seconds if it wasn't protected. Now, the problem is that if you try and protect it in a ball, like the mRNA vaccines, they're in these lipid nanoparticles.
Mm-hmm, mm-hmm.
The smallest lipid nanoparticle that you can make is about 50 nanometers in size due to the curvature. You can only make make a bubble so small, if you can imagine that.
Mm-hmm.
If I would vaccinate you with that, the only place that would go is into my liver, because we are like a leaky, porous pipe. We have very small holes. And for example, in the bloodstream, the size of the pores in the bloodstream is about 8 nanometers. Well, something 50 nanometers is not going to cross. The lymphoid flaps in my skin, if I want to inject into the skin, are about 20 nanometers. So a lipid nanoparticle won't do the trick. We can't encapsulate it.
Mm-hmm, mm-hmm.
So therefore, how is this thing protected? So it turns out, going back to this wonderful piece of chemistry, this artificial atom. So this really is, people are getting, where this isn't, human trials, they're really getting some Star Trek stuff here. This weird little crystal structure, why is it so weird, is it creates a field around the particle. So I'm not saying it's like the shield that Israel's got, but it's pretty close to it. Let me put it that way. So out to about 5 to 7 nanometers, it creates a quantum field around it, which distorts the ionic structure and the structure of water around the particle. And enzymes don't work on this particle.
Wow.
So I can take this thing and I can put this thing into me, and proteases will not eat it. In other words, so it's protected actually by a quantum field. You can measure it and everything else that we have done. That allows for the delivery. The final piece, why microneedles? So as I said, nobody has been able, the big problem with T cell therapy is you have to have the T cells in the tissues. 99.9% of all the T cells in my body right now are in my tissues. If you put them, someone's done the calculation, if you brought them all together, we'd have an organ the size of a pineapple. There's that many T cells in us, but they're spread throughout the entire body. So if I do an mRNA vaccine, hugely controversial, but yes, it does generate some T cells. But those T cells are in your blood. They do not migrate into the tissues. They are useless. You have to generate what are called tissue resident T cells. You got to have them. You got to have them there. I mean, if I want to prevent dengue, what is the point of having some T cells float around in my bloodstream when I get hit by a mosquito in my skin? Right. It's going to be, the infection's done, whatever it is. Everybody knows to prevent flu or COVID, anything else, you need tissue resident T cells. So I would love to say we were smart, but we discovered that if we vaccinate into the epidermal layer of the skin, so imagine the skin is the keratin layer, then there's a very, very thin layer just under the surface called the epidermal layer, not the dermal layer, the epidermal layer, and that's fluid. There's fluid movement in that. And that contains the highest density of dendritic and antigen-presenting cells and Kupffer cells of anywhere in the body. There are more lymphoid cells in your skin than anywhere on the body. Okay. And it's obviously it's there because, well, that's where infections come in. They come in through our skin and membranes, whatever it is. So what was discovered, and then other groups have now confirmed, is if you vaccinate or you inject into and you vaccinate these skin dendritic cells, not the ones in your lymph nodes, but the ones in the skin, they migrate up to the lymph nodes, but they get the homing signal. Because the body had to figure out, well, what's the point of me having an immune system in my skin if I wanted to protect my lungs? So the T cells generated by epidermal vaccination generate what are called tissue homing characteristics. So they go throughout the body and they go into your liver and they go everywhere and they do that protection. The only way, as we know, possible is that's the signal that happens there. So that's why microneedles then had to come along, because in order to vaccinate into 300 microns beneath the surface of the skin, you can't do that with a syringe, let's put it that way. So microneedle technology came along, which allows this to be feasible. And there's a number of devices that we use. Some of them are dipped patches where the vaccine is just dried on and it gets into the skin. That's the Sozano technology, which we bought up in Fremont, California. Other ones are hollow needles, which are very small, but obviously they hurt people and they don't like them. But the final piece to this then was to generate tissue resident T cells that will go off into your lungs and your liver and everywhere else and do the protection that you require. So I think, as you can see, my career is sort of led here. I mean, started out in inorganic chemistry and biochemistry to generating, the first people on the gold nanoparticles in terms of that. And then, of course, in the end, of course, we took glycobiology and learned how to coat. That's what it basically was, is learning how to coat these particles. And that was the big breakthrough on nanomedicine, was to make water soluble ones. So originally we made these so they'd be water soluble, because the pieces of gold obviously are not water soluble.
Right.
And that. So in the end, we've got a very exciting means. Let's put it this way. What we can do is, for any peptide, foreign peptide or cancer peptide or whatever it is, we can turn on a T cell army against that. Okay? Tissue resident CD8 T cell army. We don't worry about autoimmunity because, in the sense, we can only turn on things that you already have. Let's put it that way. In other words, we can't, like an autoimmunity in terms of IgG, where you generate an antibody against something you shouldn't, that's a bad thing. Otherwise, because you can generate new things. Here, we're turning on things that you would turn on anyway. Now, obviously, we can take a selection, because obviously there's going to be certain bad guys out there, potentially, whatever it is. So it's really, we call it, it was always the holy grail for vaccinology, was to turn on the cellular immune system. That's the Achilles heel of viruses. So we prime somebody against dengue or against yellow fever or whatever it is. T cell army is there. They are already in the skin. We know they go there. They'll home to the skin, whatever it is. You get hit with the old mosquito bite, which is just a few viruses, by the way, and you're annihilated. So we're in clinical trials around the world, and in the phase 2, mostly now phase 2, phase 3. We did all our phase 1 trials. Fortunately, most of it during the lockdown and everything else.
Right.
But we've got obviously a very, very important dengue vaccine.
Right.
As you know, there's a huge global need for one now. And I think you probably saw that the Dengvaxia by Sanofi has now been removed from the market.
Yeah.
The only one on the market is by Takeda, and the jury is out on that. But
Right.
that's not going to deal with 3.8 billion people because of the mass. So these are synthetic. I could make 10 million or 100 million, whatever it is. They're all made in a reactor. There's no live components. They are, I would call them one of the largest synthetic compounds. You can write a chemical formula down to the last hydrogen, nitrogen for our vaccine. They are simply a chemical.
That's going to be
So
useful for CMC.
yes, in other
with
words, and you
something.
can make the, yeah, yeah, you can do that. So, and of course, your question here is yes, they have, and they also get around a lot of the other problems. You know, you probably heard about these, what are called imprinting problems.
Right.
So if you vaccinate somebody against Omicron and then you go against Wuhan, blah, 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 it still wants to do Omicron, you know, type of thing.
Yeah.
So it's imprinted. That's because the body's responding in the context of an infection.
Right.
And what we're doing here, there's no infection.
Yeah.
In fact, there's no inflammation. It just simply goes in, tells T cells. And once you, by the way, you don't really have to do anything. Once you turn the T cell on, when they're called naive T cells, they already have specificity. Once you turn them on, it's all programmed. It just goes on. You don't have to
Right.
do anything. They just become.
Right.
So it's wonderful.
Yeah.
Um,
Thomas, because you mentioned dengue and you've talked
mm,
about, again, you can look at the portfolio here on the
yeah,
website and you have, chikungunya in there and yellow
yeah,
fever and a bunch of the, Peter Hotez's neglected
yeah,
tropical diseases.
yeah,
But you do have two parts of the portfolio, one beta coronaviruses and this pandemic influenza. Could you say
sure.
a few words about the pan stuff? Because I mean, the pan vaccines. I mean, that's
Yeah,
clearly. You mentioned obviously you have a holy grail of
sure.
immunoquantum glycobiology here.
Yeah, yeah.
Talk about this other holy grail because we all want these pan vaccines to get here
Okay,
for.
so a pandemic is very interesting. Is first of all, I very clear on the concept of a pandemic. A pandemic is when a new viral something or another spreads around the world globally very, very quickly. And
Right,
what it says is there's no general immunity in the population to that virus or that virus strain. So if you take the 2009 pandemic, flu pandemic, the Mexican or swine flu, whatever you call it. We know the
right,
index case. The guy named Edgar Hernandez, I have to say, in Perote, Mexico, in a kindergarten, was the first person supposedly on this earth to come down with that. Okay. Within weeks, that pandemic strain of flu spread around the whole globe.
yeah,
And if you look at this, it doesn't look like it went on airplanes. I don't know if they all had phones or whatever it is, but it moved probably in the air or whatever it is. Now, what's important, and of course, what that means is whatever was generated at that time, there had to be no immunity to it out there in the global population.
right,
Because otherwise, Argentina wouldn't have got it, or Brazil wouldn't have, if you see what I mean. But it
right,
went across to all of these countries. So a pandemic means something new has evolved in which there is no previous immunity to. So that's a hard one to get your concept around. Well, everybody's got flu. Why wasn't there any flu? Why wasn't there cross reactivity with the flu that people had prior to February 2009? But there
yeah,
wasn't. So pandemics are extremely interesting. It's one of my favorite topics, I have to say. There's a pandemic, global pandemic, approximately every 50 years for influenza A. Influenza B, you don't get pandemics, but you get them. So we had 1918, the
hmm
Spanish flu. And then you had it in 1949, right at the end of World War I. We didn't use the word pandemic yet, whatever it is. We had epidemics of Asian and Hong Kong flu and things like that
hmm
interveningly. And then we had the next pandemic in 2009. And if you go back in history, pandemics and flu occur about every 50 years, all the way back to the people have looked back to 1700s. Okay.
Mm-hmm.
So it's a very, let's call it immediately, you can say that's a rather generational thing, isn't it? You certainly, well, okay, why is flu totally changing the way the human race revolves around it approximately every 50 years? So another way of looking at it now, I had to be the descendants of Spanish flu. So I would have had to been born I can just tell you, between 1918 and 1948. Okay. If I was born during that period of time, I would have been exposed to a descendant of Spanish flu, because that was the pandemic. Just like the easiest way to look at it right now, the only pandemic strain circulating in the world is influenza A California, which generated in 2009. Okay, let's put it that way. So everybody born now is exposed to what's called a pandemic strain. So they're all A California 2009. Between 1918 and 1949, they were all to A Brunswick, whatever it is, which is the Spanish flu. And from 1949 onwards, they were all to A Albany, whatever. So on top of all these little tiny things, the pandemics are fixed in 50-year cycles. So you then have to ask, well, how does this all of a sudden, because how did this flu, what was it the flu virus lost? Because it has to be a loss, remember, because it's now going to go out there. And I've got, put it this way, everybody born between 1949 and 2009, we've all been exposed to flu. So in 2009, flu appears and it goes around the world, which means I no longer had immunity to it. So it's not like it gained something, it lost something that I had, and now I no longer have immunity to this new strain. So it's therefore, because we worked a lot, we've presented at the CDC, at the FDA, and NIH on this. This is generally accepted on this. So therefore, what we set out to do, therefore, is look for the loss, in other words, of this. And this has to be, therefore, something. If you could imagine, right now, if you went on the internet, you'd probably find 150 strains or something of influenza 2009. There'd be A Wisconsin, A Michigan, all the little substrains that get deposited in. But they still all have to have the same loss. So what we discovered, and which was an extremely important discovery, is that a flu is what's called a negative strain, a negative RNA virus. It's an RNA virus, but it's a negative sense. So when it goes into you, it can't make, I talked earlier, any of the proteins, because it first has to go from the negative sense to the positive sense.
Okay.
But what we discovered was that flu had a ligandome signature immediately upon infection. And what we then discovered, of course, is that the negative strand of segment 8 of flu codes for peptides. And those peptides are the things that change every 50 years. So they're constant for 50-year cycle. So there was a set of immune peptides produced by every flu virus in existence between 1918 and 1949 was the same. And then between 1949 and 2009 was the same. And in 2009 onwards, it's the same. So that is the pandemic element. So the trick, therefore, is decide, well, what's going to be the new pandemic element? Because if we can vaccinate people against the change, then they'll already be vaccinated against the next pandemic, if you see what I mean.
Right.
So therefore, we've been monitoring. We monitor every flu strain deposited. And we monitor for this pandemic element. And there's two choices that we have. Either the pandemic element from a previous pandemic is going to reappear. So you could say, okay, nobody living in 2009 or in a few years, but wait, by the time 50 years is up or in 2025 through 30, there'll be nobody living that will have immunity anymore to Spanish flu.
Right.
They're all dead.
Yeah.
Okay. So this would be a pretty smart thing for the old flu virus to do, is to come back and recreate the pandemic element from Spanish flu, since nobody has immunity to it anymore. They're all dead. Another way of looking at it, I used to try to explain this. I have 100 people in the room, and flu has its way. And in year one, it's got 99 victims. And after 50 years, it's probably searching around for a few victims. It's got, now, who am I going to infect, whatever it is? So flu seems to do this, not by genetic drift, but all at once. It just sort of changes it all at once. So what we've observed is, we're monitoring this, is that we are seeing, believe it or not, because we can see these, we are seeing increased cases of descendants of Spanish flu now occurring around the world, isolated cases. So this is supposed to have gone away, but it's not. It's still there. We can actually see this. The other way of looking at it is so on our current vaccine, what we've included in that vaccine is elements would cover you against all known pandemics. So we've got elements in there that will give you the immunity if you were born in 1918. We've got elements in there that'll give you the immunity if you were built in 1951 and onwards. So we're giving you, in our vaccine, uniquely, everything for the last 123 years or so. Anything that appeared in the last 123 years, our vaccine will immunize you with. So that's the first way of looking at it. So that truly is a pandemic. It has nothing to do with thing. So the second thing is, there's an odd relationship. Before every pandemic, there was an outbreak of equine flu. Now, we're all talking about avian flu, but if you look back, the words, and they even say, if you think about World War II, 1918, there was an outbreak of equine flu, and there's all kinds of theories that it came from the horses. And in other words, we used to be a horse-related society. And that's absolutely documented, whatever it is. And so there's a controversy of whether or not there's a jump from avian birds into humans. On the other hand, there's also a possibility that there might be a jump from horses again. In other words, as the reservoir. So our vaccine, I thought, is quite clever. We're also vaccinating people against avian and horse flu. So because they contain a different element. So they contain and if that element, for example, so the horse element, the pandemic element in a horse, is different than human. Okay, let's put it that way. So therefore, we figured, well, if it jumps from a horse, it's going to be called pandemic element 93. Currently, we're on pandemic element 87. Well, you'll all be prepared for that. So in terms of that, our vaccine here will generate this commonal, it's called the birth cohort immunity, but that's the thing. And we're building in a horse, equine, and of course, right now there is reports out there about equine, and of course, people are worried about avian flu. And again, it could be avian flu, whatever it is. So again, interestingly enough, this is what's called, I can't remember what name they use, because this is all coming from the negative strand. So this is stuff that doesn't even come from a protein that was never made. This is an immune element of flu. Now, I did mention, I'll just say in the end, influenza B, interesting, doesn't contain any pandemic elements. And by the way, there's never been a pandemic influenza B pandemic, whatever it is. Now, the end, we did look in SARS-CoV-2, and interestingly enough, we found a pandemic element in SARS-CoV-2. In other words, and we call it ORF 100, and our current COVID vaccine, which is a pan. It was actually developed against SARS-1. That's where we were developing it. Then SARS-2 came along, and it works against SARS. It's pan coronavirus, but it actually will protect you against the pandemic element of coronavirus. And there are some people believe there have been coronavirus pandemics. It's just that we didn't recognize them as such, if you see what I mean.
Right.
So I'm hoping that our current corona construct, which is going into phase 2, will also be considered a potential SARS, in other words, in other words, if there's another, because there was a pandemic, however you look at it, the SARS-CoV-2 with all of the definitions of pandemic. It spread around the world within weeks, blah, blah, blah. And it did all of these things. And now you got lots of substrains, just like flu. There's a billion substrains of A. California. So it's exactly the same.
Yeah, no, it's no, that's amazing. Really an entirely unique way to look at because we just think of the virus, but clearly these
Oh, I think they got mobile phones.
fingerprints.
I think they got mobile phones. In other words,
Yeah, it's
yeah.
rampant.
Well, yeah. I look at I just get people call me crazy, and I just it's I'm because not only that, we got to remember what happened with in 2009, the circulating strains in 2009 that were there vanished. Now it just got replaced. It's not like I'll take over a little bit of the world. All the ones that were previously circulating vanished within weeks. It's absolutely astounding. In other words, so that's why everything circulating now is derived from Edgar Hernandez. Which is, except for a few things below. I think there are a few things that survive in very outlandish communities, which are very isolated. Let's put it that way. But we won't go into that.
No, understood. No, this is Thomas. This is an amazing journey. I can look forward to continue to see the evolution of this as these really elegant tools that you've developed get through clinical trials. I'm gonna love to see what happens. Obviously, I sit in the here in Philly in Immuno-Oncology Central, so be interesting to see where all the these concepts ultimately are taken with regard to that whole area and the sort of our understanding of what it means to go after these unique antigens and so forth in the oncology field. But no, it's great stuff, and I wish you and the team there at Oxford the best with it all. And hopefully we can do a follow up with you next year as some of these things come through the pipeline. Please.
Anyway, just going to say, by the way, all of our ligand domes are done just down the road from you in Doylestown, Pennsylvania. As we have Emergex USA is based just down the road, and we all fly in through Philadelphia usually there. So that work is all done by some brilliant this is all high level mass spectroscopy and things like that. But that's all done in the United States, and obviously we obviously work with the government labs to get a hold of all these odd strains and things like that, because fortunately we only need to grow them once, and then we know what the signature is, and then we never have to do it again. So, but we still have to do it once. Okay. Anyway, no, it's been a great pleasure. Going through that, and I think we sort of did tie the threads together.
Absolutely. No, it's really enjoyable. And again, for everybody that's going to be listening to this particular episode of the show across the podcast networks or watching on our YouTube channel, again, you've been spending time with Professor Dr. Thomas Rademacher, CEO, co-founder, Emergex. Tom, I want to again thank you for taking the time out of your schedule to really go in depth here, and obviously, thank you for what you do. And as we like to say on our show, thanks for helping to create a better tomorrow for literally billions of people out there via the development of these technologies. It's a really, really great story and rooting you on.
Okay, thank you. Thank you very much.